Good morning. Good morning. It's 10:15 a.m. Eastern Standard Time in the morning on Thursday, October 5th, year 2023. My name is Mark Kulak. This is Houstonic.live, a research archival show in our fifth year right now. And we're using Live 2 today. We need to, out of necessity, maintain a few different YouTube accounts. And this is a new series, 245, we're going to call it. And in this series is going to be a reading of a gold mine of an article. This article is long enough that it could be a book effectively on its own. The article is published on the website by whatisbiotechnology.org. I put the URL into the show notes for what is right now only on Live 2. And we're just going to do this reading live. There may be a few times during the reading that I will need to take a pause and maybe find a video so that because there's many names involved in this very lengthy multi-part article. And it's so easy to mispronounce some of the names. So there may be some times that I want to take a pause for that. The article itself is broken up into, I think, between 9 to 12 parts. And based upon how fast we can read it, each part may turn into its own episode. And then from there, we will make a playlist. The article title is A Healthcare Revolution in the Making, the Story of Cesar, Milstein, and Monoclonal Antibodies. So, why are we focusing so much on monoclonal antibodies right now? We just did a series called the Antibody Patent Paradox, a name that was lifted from a 2023 Yale Law Journal article about the evolving challenges with the patent system and how it works with monoclonal antibodies. And although the article doesn't say it, it's really about the challenges of patenting proteins in general. There is still much unknown. I should say, the more that is learned about proteins and antibodies, the more we realize, or the industry realizes, it doesn't know. In fact, there used to be just very broad sets of antibodies which were appropriate for the time based upon the state of the art of science at the time. And so much has been learned about them. So much has been learned about what we don't know, I should say, at least highlighting what we don't know, that what used to be fact has now effectively turned into mythology or science fiction. The antibody patent paradox, although it may sound as if it is about trying to facilitate companies protecting their intellectual property, I believe it highlights a much more than just that. I posted a 3-minute video yesterday that I only found thanks to a reference from Jonathan Cooey, Giga Biological. You can check him out, gigahomebiological.org. He streams on Twitch, where one of the two Nobel Prize, 2023 Nobel Prize winners for, I don't know what the category, science or biotechnology, Drew Weissman, is presenting in an October 2022 event where he makes a comment that mRNA is effectively superior and very advantageous over monoclonal antibodies because of the length of time, the amount of equipment, and the cost of manufacturing monoclonal antibodies. And while to some extent I'm sure that there's some truth to that, much has been said about the complexities of monoclonal antibody manufacturing. Much has been invested into, there's been significant research into what other methods can be used, such as plant-based. That is a company that Michael Callahan, in fact, has been participating in that, University of Alabama. There's a scientist up at Winnipeg, Gary Kobinger, who was, so he goes along with much of the main line. He is also a little bit of a dissident in some ways. He's an interesting dude, Gary Kobinger. Also happened to be peer of both Heinz Feldman, Francis Plummer, rest in peace, Guangao Qiao, the accused Chinese spy. So that's three heavy hitters that he worked with up in Canada. Now, what we are observing is potentially a battle, if you will, between mRNA medicine, a way of being able to, there's ways to manufacture that, and then the whole cycle. The mRNA supposedly it causes a protein to be manufactured, expressed by the cells. Is it a foreign protein? Is it is it a misfolded protein? Lots of discussions there, and then it's supposed to turn into the body producing an antibody to it, which I believe it does. Though there's much that isn't typically discussed about the variability of the protein expression and so on and so forth. Now, there is a very different series of, first of all, the way that is tested or approved by the FDA is efficacy. Efficacy just means that they were able to test for the presence of an antibody. In other words, it's effective. Doesn't mean it's actually going to protect you. It means that it will provide something that is a correlate of protection. It's a little bit like, if you were to give someone who's driving a car, a single piece of tissue paper, and say, put this in your front pocket, it effectively does provide more protection should that person be in a car accident. It doesn't really save them, but it is effectively giving them more protection. And that's how mRNA is measured. That's how many traditional viruses have been measured. Are there antibodies? It doesn't really do a good job at telling you how many. That's guesstimate. Are there any? And if there are, it's effective. Boom. Meanwhile, with monoclonal antibodies, how is efficacy tested? Well, it's the antibody's already there. You clearly know it. It's it's already in the little vial. It's being injected. So you can't test it. So how do you test monoclonal antibody efficacy? Challenge experiments. Do people who have certain disease have it already or are exposed to it in some type of controlled environment? Challenge experiment. Do they recover? Do they live longer? Because you can't test for the antibodies because that's the medicine itself. Hence, mRNA has a dramatic advantage over monoclonal antibodies with respect to how it's tested. Matter of fact, noteworthy that there's another thing that tests very similar to monoclonal antibodies, antivirals like remdesivir. Also, the patenting, when you're patenting monoclonal antibodies, you're patenting the antibody itself, of course, and the structure. And you have to make estimates about how the structure impacts, the ability to fight or cure a particular type of cancer, recover from a certain disease, whatever the situation is. Whereas with mRNA, again, all you need to do is patent the sequence. And you can even open source the platform. So you can see that these two are at war with each other with respect to intellectual property, with respect to much. And this war may even have manifested itself into being represented by political movements. Is it possible that some type of political ideologies have actually adhered to one technology over the other? Anyways, as I've said before, since I've started doing the research after COVID started, I didn't think COVID was going to be a big deal, like when I first heard about it back in early 2020. Boy, was I wrong. I kind of poo-pooed reading much about monoclonal antibodies. I don't know why. The topic just didn't present itself as fascinating in such a fascinating way. In fact, when I was reading the book that was published the week after September 11th, 2001, called Big Shot AIDS Vaccine, I actually skipped over the antibody chapters because it just seemed irrelevant. I was missing the point. It's not mRNA versus traditional vaccines. It's really mRNA versus monoclonals. And Drew Weissman, whether he was just being stupid or not, or maybe it was a little bit of a hidden white hat moment to put it all on display in 2022. That was the first time I've heard someone say the advantages of mRNA over monoclonals. Until then, it's always been mRNA versus the old traditional vaccines. Anyways, I just wanted to spend a few minutes to highlight here the many paths by which we are becoming aware of the importance of monoclonal antibodies, the importance of manufacturing proteins in general, and testing the impact of proteins that have been manufactured and in different ways that they've been manufactured. Not to mention, the only way monoclonal antibodies can be made is through them being expressed by cells, manufactured by cells. Oftentimes, those cells are connected with cells which have been cloned from years and years and years ago from aborted pregnancies. So there is that issue as well. Also, and you have to use those cells because those cells are humanized. We're not just plug and play. You can't just put mRNA into a mouse cell or a hamster cell or a monkey cell, and it's not going to be expressed the same way a human cell will do it. We know that now. You need human cells. And another convenient way to get around that is to just inject the mRNA directly into a person. So there's that as well. And maybe someday that will bear fruit, whatever the case. So with that set up here, let me just do a quick check-in to make sure all the audio is working and everything else. How many do we have here? 9 watching. Excellent. Pop out chat. We have a couple family appointments today. So we will not, again, so I apologize again to everyone as our family, evolves. I just have to take, if I get an hour, I just have to take the hour when it's there. And that's it. I could spend the first 45 minutes of it going through all the notifications, and then I lose my hour. So I just have to go for it. And I know it impacts views and visibility and all that. It is what it is. Okay. Hello, End of Days Prophet. Yes, they have to be. Well, the thing is that when proteins are expressed, end of days, when proteins are expressed, multiple things impact the type of proteins or antibodies that are manufactured. Obviously, the recipe, we heard about this, the recipe, the template described by the mRNA is, of course, a big part of it. Another big part of it, though, is one's own DNA. Someone's own DNA impacts how a protein is created. The protein is not going to be identical between person to person or even between cell to cell. And if you wonder, well, how does the DNA come into play of the person, of the host, right? Each ribosome has been constructed in and of itself based upon the genetics and a bunch of other epigenetic conditions. Ribosomes are not all exactly 100% identical. They even vary within cells themselves. So as there's this galactic variability in proteins that are expressed. As I've said before, the spike protein, that phrase itself is a lie. There is no the spike protein. There's a category of a protein which can be manufactured. However, the specifics will depend upon the individual. They will depend upon the cells, the muscles which are doing the expression, which can vary between shot to shot. They will vary based upon epigenetic conditions, such as diet, age, and so on and so forth. So the spike protein is a lie. There's a category of proteins which are being expressed. And the variability could be massive between people. This is not being considered. And it's largely unknown which factors are going to impact it how. It may be that needs to be determined. Immunogenicity of direct mRNA, though. Well, I don't know if you're talking about an autoimmune response, Kim. But it's some of the people that I'm working with here, I'm so blessed to be working with, such as Sam Eagle or Regular Guy, are helping me take this to the next level. All right, we found some great correlations. We got the ball rolling. We knew where to shine the flashlight, but I can only take it so far, right? Like a football team, some people can run really fast. Some people, will get you 4 yards every single time they get the ball, no matter how bad your offensive line is. Different people have different strengths. And right now, I'm fortunate enough to be teaming up with a few other people to help out. Taz says, cookie cutter medicine isn't real. Generally speaking, yeah. I mean, categories of medicine are usually, will work similar in most people. Most people will have reduced inflammation if they take an anti-inflammatory. Most people will respond certain ways to opioids and so on and so forth. But we're not all identical, that's for sure. But on a molecular level, this is really where it's key. On the molecular level, just saying the spike protein is a lie. That's I call it the spike protein deception. Okay, so let us start reading the article here. Just in case you're curious, on the menu, these are the different sections. Introduction to the exhibition, Milstein's early life and work, Milstein's early antibody research, the making of monoclonal antibodies, the patent saga. Yes, antibody patent saga boils all the way up to the top. From Milstein's laboratory to the world, the early commercialization of monoclonal antibodies, the first clinical applications of monoclonal antibodies, the legacy of Milstein's work. And I don't think I will spend sections, rereading the glossary and the Milstein timeline and so on. But so let's just get into it here. I'm a little bit gassy right now, so sorry about that. Healthcare Revolution, the story of Cesar Milstein and monoclonal antibodies. Collated and written by Dr. Laura Marks. Zoom in a little bit here. Today, 6 out of the 10 best-selling drugs in the world are monoclonal antibody therapeutics. This was written about 3 or 4 years ago, I think. So think about that. Actually, this was written almost 10 years ago right now. So as of 10 years ago, 6 out of the 10 best-selling drugs in the world are monoclonal antibodies. That's a big market. Christy, great question. I think based upon the last date of revenue being 2012, I'm going to take a wild guess that this is 2012 or 2013. I should download and archive this article, all these articles. In fact, I probably will later today. I've been meaning to do it. There's great photos in here that I can't get anywhere else. And if you don't think it's important, I know that some people are in New Orleans this week with some Dr. Mary's monkey research. The polio vaccine issue, contamination SV40, does involve a Hillary Kaprowski of the Wistar Institute. Okay, let's agree on that. Hillary Kaprowski is then described as a lucky and wealthy Polish man who family just happened to have the foresight to scoot out of Poland just before the Germans entered. And then he was lucky enough to go to Brazil and work for 4 years as a refugee for the Rockefeller Institute on yellow fever vaccines. And I believe he was working with Alice E. Moore at that time. Now, I don't know about you guys, but to me, that sounds like the story of what an actual Nazi scientist would have done. But we're not going to go there just now. This article, not this section, but this article does mention, in fact, a huge part of the monoclonal antibody story is also Hillary Kaprowski. And in fact, he and the Wistar Institute effectively stole the patents from Britain. In the 1970s, 1980s. And then Kaprowski went on to be the founder of Centocor. Centocor, which was acknowledged by some to be the first monoclonal antibody company. And Centocor was represented in a 2010 lawsuit by none other than Les Weinstein, father of Eric Weinstein, who tells Palantir how to invest their money in monoclonals, and Brett Weinstein, who introduced Robert Malone to the world, effectively. Hillary Kaprowski is also the mentor of both Stanley Plotkin and Dave Weiner, both senior people at Inovio. The monoclonal antibody story is heavily connected to the world of virology and vaccines, deeply connected, because the world of virology and vaccines is about getting cells to do crap, to express proteins, and to see what happens under the guise of emergencies. Anyways, patents, Hillary Kaprowski, are connected to this. And if you think Kaprowski is just a guy who, had some oopsies manufacturing polio vaccines, you are way wrong. That's what I thought for a while too. That's why there's books written about that. But the real Kaprowski story is wild. It's really, really wild. So with that said, I needed to just emphasize how this ties in to what seems like other unrelated research. And the cell cultures manufactured for Hillary Kaprowski, who did that work? It was Alice E. Moore, who also ran cell cultures for the Special Virus Cancer Program. And that's the reason why, amongst many, Alice E. Moore, and I still don't know what the E stands for, is not on Wikipedia. There's no Alice E. Moore's Russian encephalitis book for a reason. All right, so let us begin again. Collated and written by Dr. Laura Marks. And I'm not going to be looking at the comments for the next however long until I finish this. Today, 6 out of 10 of the best-selling drugs in the world are monoclonal antibody therapeutics. One of these, Humira, which is a treatment for rheumatoid arthritis and other autoimmune conditions, was listed as the top-selling drug across the globe in 2012 with a revenue of US $9.3 billion. Based on its current performance, many predict the annual sales of the drug will surpass the peak sales of Lipitor, a treatment for lowering cholesterol that is the best-selling therapeutic of all time. Currently, monoclonal antibody drugs make up a third of all new medicines introduced worldwide. Noteworthy that my Uncle John passed in his late 50s, I believe. Might have been 60, but I think it was late 50s after he started taking that drug. Suddenly, doctor says, oh, you're a little overweight. You got to take this cholesterol reducer. Next thing you know, he's got liver cancer. So, and that story is not unique. Monoclonal antibodies are not only successful drugs, but are powerful tools for a wide range of medical applications. On the research front, they are essential probes for determining the pathological pathway and cause of diseases like cancer and autoimmune and neurological disorders. They are also used for typing blood and tissue, a process that is vital to blood transfusion and organ transplants. In addition, monoclonal antibodies are critical components in diagnostics, having increased the speed and accuracy of tests. Today, the antibodies are used for the detection of multiple conditions, ranging from pregnancy and heart attacks to pandemic flu, AIDS, and diseases like anthrax and smallpox released by biological weapons. Beyond human healthcare, monoclonal antibodies help detect viruses in animal, livestock, or plants, prevent food poisoning, and investigations into environmental pollution. Monoclonal antibodies are indispensable in so many walks of daily life thanks to their ability to target a single type of cell. Produced in the laboratory, these antibodies are derived from naturally occurring proteins made by the body's immune system to recognize and fight foreign invaders, such as bacteria and viruses. The antibodies are generated through the fusion of a myeloma cancer cell with spleen cells taken from an immunized animal. Yet the story of how these unsung microscopic heroes came into the world and helped change healthcare remains largely untold. Moreover, their significance was largely overlooked at the time of their creation. The journey of monoclonal antibodies all started when an Argentinian immigrant named Cesar Milstein, and last name spelled Milstein, Milstein, one L, Milstein, arrived at the Laboratory of Molecular Biology in Cambridge, the same laboratory where Francis Crick and James Watson discovered the structure of DNA in 1953. It was to be here that Milstein, together with Georges Kohler, K and then the O with the two dots on top of it. I apologize. I have some German ancestry. I should know what that thing is. Kohler pioneered the seminal technique for the production of monoclonal antibodies in 1975 and showed their clinical application for the first time. This exhibition of the life and work of Cesar Milstein provides a window into the world where monoclonal antibodies were first developed. Showing Milstein's notebooks and writings for the first time, this exhibition provides firsthand the complexities that were involved in the creation of monoclonal antibodies and brings to life the many challenges scientists face in devising a viable biotechnological tool and its application in healthcare. Transforming monoclonal antibodies, which started life as a laboratory tool, into something that could be of use in the outside world was neither straightforward nor inevitable. From Milstein's papers, we learn firsthand how the newly created monoclonal antibodies spread from the confines of Milstein's laboratory in Cambridge to scientists across the world, and where then were then adapted for clinical applications. They highlight the logistical difficulties Milstein and his team faced in transporting monoclonal antibodies to other laboratories, and the fact that other scientists initially had little idea about how to grow and maintain the antibodies, let alone any idea what purpose they might serve. Strikingly, initially Milstein had very few requests for monoclonal antibodies. By 1977, however, he was being inundated with requests for samples and had to search for outside support in the distribution process. This was to pave the way to the earliest commercialization of the technology with the help of Sera Lab, that's SERA-LAB, Sera Lab, a small British company set up to supply animal serum reagents to the scientific community. The relationship between Milstein and Sera Lab illustrates the process of technology transfer in biotechnology during its formative years. All of this was done with little public fanfare and no venture capital or government support. Yet, the collaboration between Milstein and Sera Lab laid the foundation for the wide-scale commercialization of monoclonal antibodies. The exhibition also offers a way of understanding why the original technology developed by Milstein and Kohler was not patented in Britain and instead formed the basis of patents taken out by the Polish-American virologist Hilary Kaprowski and his team based at the Wistar Institute in Philadelphia. And when you see, and I'm telling you, everyone, when you see Hilary Kaprowski, you should be thinking of Dave Weiner and Stanley Plotkin and Inovio. This is a narrator's note. And the Eros Tuberculosis Global Vaccine Foundation, and so on and so forth. And Robert Malone. Let me repeat that sentence. And this is about patents also. And again, as the antibody patent paradox reading highlights, the patenting of antibodies is turning out to be effectively an unresolvable nightmare. But if you can just have them produced via mRNA, you can bypass all of that. Hence, one of the biggest industries out there is effectively in and of itself, and I'm not saying that it's all good, but being attacked using a different technology, which plays a big part in bypassing the patent system and the antibody patent paradox, which is facing monoclonal antibodies. Can you see it? Can you see why there are so many people who don't want you thinking and talking about patents when it's possibly one of the foundational reasons why certain technologies are being used right now? Have to reread that paragraph. The exhibition also offers a way of understanding why the original technology developed by Milstein and Kohler was not patented in Britain and instead formed the basis of patents taken out by the Polish-American virologist Hilary Kaprowski and his team based at the Wistar Institute in Philadelphia. The latter were thus the first scientists to be granted patents for monoclonal antibodies. Generating major controversy in the late 1970s, the patent story told in this exhibition, in other words, this set of articles, reveals some of messy business of patenting research science and the implications this holds for those working in both the laboratory and the commercial world. It also provides some insight into Milstein's very early efforts to demonstrate the practical application of monoclonal antibodies. He and his colleagues paved the way for the use of monoclonal antibodies as tools for the purification of natural compounds for drugs and as the reagents for blood typing. Their work also demonstrated the use of monoclonal antibodies as probes to investigate the pathological pathway of neurological conditions and a wide range of other diseases. People who are focused on the brain, on neurological issues, should be very much interested in this topic. Really interested. This paved the way to the adoption of monoclonal antibodies as diagnostic tools and as an invaluable platform to move towards personalized medicine. And I'm going to read that sentence again twice. This paved the way to the adoption of monoclonal antibodies as diagnostic tools and an invaluable platform in the move towards personalized medicine. However, this is narrator's note, as more is learned about them, they realize that there is a patenting nightmare. A nightmare which could potentially be resolved, potentially, with mRNA. If the body just makes its own. The final part of the exhibition shows how Milstein encouraged the use of genetic engineering to improve the safety and efficacy of monoclonal antibodies, thereby enabling their use as therapeutics on a large scale. So we're not going to read the first section here. Celebrating the first publication of monoclonal antibodies. It's now 41 years since Cesar Milstein and Georges Kohler published their technique for producing monoclonal antibodies. And this is effectively a celebration. There's also a film that's linked to here. I'll check that out later on. I haven't clicked the link yet, so I don't want to do that on the air. And what I will do is I'm going to embed the 3-minute video that I made yesterday. It's important because our new Nobel Prize winner, Drew Weissman, is going to highlight to you what he knows about the benefits of monoclonals over, of mRNA over monoclonals. And then after that, I'm going to sign off, and we'll do our part 2 when we start our first section. This is going to be a lot of fun. It's going to be a lot of learning. This is not a series for everyone. This is a series for the researchers, the people who want to watch the shows that are at the lead right now, because we're going through this stuff together. I appreciate everyone's support. God bless. Have a great day. I'm really, really excited about this stuff. I'm really getting pumped about where we're going here. So let's just play this little clip, and then I'm going to sign off right.
I know that's all just lies. So general health. Get outside multiple.
Okay, here we are. Sorry, I had a video playing somewhere else.
I'd like to start off. And this goes back to conversations I had with leaders in the field, people like Gary Nabel and Tony Fauci and numerous Nobel Prize winners. Early in the 2000s, when I would go to a conference and present my RNA data, and they would say to me, "That's very interesting, Drew, but when are you going to do something useful?" RNA had failed clinical trials. No biotechs or pharmaceuticals had any interest in it. And they thought I was wasting my time studying RNA. And they were trying to convince me to move on to something else. And my argument was always, well, RNA has enormous potential. At this time, and even now, the largest field in pharmaceutical sciences are monoclonal antibodies. There are monoclonal antibody treatments for cancer, autoimmune diseases, hypercholesterolemia, osteoporosis, migraines, and they keep developing more. The problem with monoclonal antibodies is this is what it takes to make them, in a factory. You start with a 50,000 liter drum full of cells. Those cells make your protein.
He won't mention the patents. This is perhaps partially true, but he won't mention the patents.
Then you have to purify that protein away from all the fetal calf serum and other cell culture components. For every new protein, you start over. The cost of a monoclonal antibody treatment is $50,000 to $100,000 a year. So that's excessive for much of America, and it's completely untouchable for the majority of the world. With RNA is made in a simple enzymatic reaction. There are no human cells. It's simply an enzyme and some chemicals. For the current vaccines, J&J uses 50,000 liter drums to make their adenovirus vaccine. Moderna and BioNTech use 100 liter bioreactors.
Note. He also called out the production problems of a DNA vaccine, the adenovirus vector, from Johnson & Johnson. Not just the monoclonals. That's interesting. I just picked up on that.
To run their simple enzymatic reaction. They both produce about the same number of doses of vaccine. For RNA, if you want to change vaccines, you want to put a new variant in, or you want to make a flu vaccine or HIV or anything else, you just pop in a new sequence and everything else is identical. For protein vaccines, for adenovirus vaccine, you start over and you reinvent everything. So RNA is fast, simple, and as we've seen, very easy to expand.